Welcome back to Biology 2210 at CNN. We are starting a new chapter in our ebooks, chapter 12, which is the beginning of the nervous system. In this first chapter, we're going to discuss nervous tissue. So we're going to discuss the various types of cells that make up nerve tissue, and we're going to discuss the basics of neurophysiology, seeking to understand action potentials and how they propagate and are transmitted from one cell to another. The nervous system is a very complex system, and you'll notice we have many chapters in the book devoted to the nervous system. But again, in this first one, we're just going to start by looking at the cells themselves. As we will emphasize many times in both AMP1 and AMP2, the endocrine system and the nervous system are very closely related systems in that they both are involved in responding to stimuli in a way to maintain homeostatic balance and coordination. They actually both respond with chemicals as well. We've already seen that the nervous system secretes neurotransmitters at synapses, such as acetylcholine was released at the neuromuscular junction. The endocrine system also responds by secreting regulatory chemicals, but in this case they're secreted into the bloodstream for delivery to a target tissue, and those chemicals are known as hormones. We will discuss the endocrine system in AMP2. So the nervous system, again, is a complex system, but its essential functions can be broken down into three basic processes. And these three basic processes are reception, perception or integration, and response. Now, reception involves simply the receiving of a signal. And this signal we usually refer to as a stimulus. And the stimulus can be internal or external. External would be things that you see, things that you touch, things that you taste. These are all external signals. Internal signals are sometimes we're less consciously aware of them, but remember, proprioception is the monitoring of the joint position and muscle tension in order to maintain balance and equilibrium. We also sense internal factors such as blood pressure, blood osmolarity, a whole variety of different things, body temperature. So there's a whole slew of different kinds of stimuli that have to be received. And we often have specialized sense organs to carry out this task of sensory reception. The messages are then that are received are then usually carried to a central nervous system where the information is processed. And by processing, we mean we make some kind of sense out of it. The central nervous system makes a decision as to what the stimulus means and how to respond to it. This is again called processing, neural integration, perception, a variety of terms really just to talking about the same basic process of figuring out what the message means and what to do about it. And then of course the final and third major function of the nervous system is to create a response. And normally that happens by sending a signal back out from the central nervous system to the body somewhere to cause an appropriate response to that stimulus in order to maintain homeostasis. Interestingly enough, there's really only two kinds of responses that a nervous system can generate, and that is it can either stimulate a muscle cell to contract, or it can stimulate a gland to release its secretion. Those are the only two mechanisms we have for responding through the nervous system. Okay, so that's a broad overview of what we're trying to accomplish with our nervous systems. Now, in terms of how the system is organized, essentially we organize the system just for the ease of study into two major components known as the central nervous system and the peripheral nervous system. And I've already mentioned central nervous system a couple times today. So what's the difference? The central nervous system is basically your brain and spinal cord, which are, as we know, protected within the skull and vertebral column, respectively. And the central nervous system is where the majority of integration takes place. It's also where we initiate the response to stimuli. So integration, perception, whatever you like to call it, and at least the initiation of response occurs in the central nervous system. The peripheral nervous system, on the other hand, is essentially the rest of the nervous system outside of the central nervous system. And so the peripheral nervous system consists of nerves, which are, as we'll see, primarily axons, which are long extensions of neurons that carry action potentials. Ganglia, which are clusters of neuron cell bodies that are outside the central nervous system. As we'll learn, the vast majority of neurons are located in the central nervous system, but sometimes you'll find neurons out in the peripheral nervous system, and I mean entire neurons, not just axons, and they're usually clustered together in structures known as ganglia. So the peripheral nervous system primarily consists of nerves and ganglia, and of course there are, as I said earlier, some specialized sensory organs as well, things like the eye, the ear, and so on. Now, our tendency to organize and subdivide doesn't end there. The peripheral nervous system contains two subdivisions itself. The subdivisions of the peripheral nervous system are the sensory division and the motor division. The sensory division of the peripheral nervous system, also known as the afferent division, is that part of the peripheral nervous system that's carrying messages into the central nervous system from the body. That part that's carrying messages into the central nervous system from the body. Afferent is a word that means incoming, and so sensory or afferent division, again, is that part that's carrying messages in, usually through nerves, either into the spinal cord or directly into the brain itself through what we call cranial nerves. We organize the sensory division even further down into what's called the somatic sensory division and the visceral sensory division. The somatic sensory division carries messages, as you can see here, from skin, muscles, bones, joints. These are things that we tend to be more consciously aware of, sensations from these parts of the body. The visceral sensory division carries in signals from the visceral organs, the heart, the lungs, the stomach, the urinary bladder, intestines, places like that, gallbladder. While we can certainly feel these things, such as in the case of a stomach ache or a heart attack, oftentimes these signals are just under the conscious level of perception. And so we're monitoring things like stretch of the lungs that automatically tell us when we need to stop breathing in, for instance, so we don't overinflate the lungs. We're sensing the tension in the heart muscle as a way of helping to regulate blood pressure and so on. So many of the visceral messages in this visceral sensory division are operating at a subconscious level. Now, the motor division, also known as efferent as opposed to afferent division, is that part of the peripheral nervous system that, of course, then does the opposite. It carries signals away from the central nervous system out to the effectors. Effector is a word we use to simply mean something that the nervous system will stimulate, a body part that the nervous system will be able to interact with and stimulate. As we said earlier, there's really only two types of effectors for the nervous system. They are muscle cells and glandular tissue. And so the motor division carries signals from the brain or spinal cord out to muscles and glands, telling them to secrete or contract. Again, we can organize the motor division into two subdivisions as we did with the sensory division. We have once again a somatic motor division and a visceral motor division. The somatic motor division carries signals to the skeletal muscles. And as we talked about earlier with the somatic division and with skeletal muscles in general, we have both voluntary and involuntary type contractions. And so the somatic motor division is that part that can carry these nerve signals, both voluntary signals and some involuntary signals, to the skeletal muscles. The visceral motor division is probably better known as the autonomic division, and it carries motor signals out to glands, cardiac muscle, and smooth muscle of the body. And what you're probably already aware just by looking at this list of effectors is the autonomic division or visceral motor division is involuntary in its action. We don't have conscious control over how we stimulate our glands, our heart, and the various smooth muscles in our body. So when we talk about the autonomic division, a lot of people think it's just a part of the overall nervous system. They don't realize that it's really just one subdivision of a subdivision. It is just the involuntary part of the motor division only consists of outgoing messages of the peripheral nervous system. So a division of a division of a division, basically. And just continuing on with this division after division after division, the autonomic nervous system itself can be divided up into two subdivisions, and you've no doubt heard these two subdivisions, the sympathetic division and the parasympathetic division. The sympathetic division, sometimes simply referred to as sympathetic nervous system, is the portion of our motor division that's involved in what's called the fight or flight response. This is the part that gets us ready for intense activity. So we often associate with things like an adrenaline surge associated with a threat or being scared or an emergency situation, but it doesn't have to be that extreme. Something like exercise will also elicit a sympathetic response because during exercise, again, our muscle activity is increasing and we need to get the body ready for that increased activity. And so the sympathetic division generally does whatever is needed in order to promote body changes associated with that increased level of activity. It's a mistake to think of the sympathetic division as being excitatory in every case. We tend to think of the sympathetic division because it's the fight or flight response. Well, it's going to excite all of its targets. But if you think about it, that's not necessarily true. So yes, it's true that during a fight or flight response, we need to increase our heart rate and we need to increase our rates of respiration, and those are all excitatory. And we also have to generally constrict blood vessels throughout our bodies to increase blood pressure and blood flow through the system. Those are all excitatory effects. But there are also some inhibitory effects of the sympathetic division. For instance, the sympathetic division has an inhibitory effect on the smooth muscle that's in the wall of your bronchioles in your lungs, the air passages in your lungs. It has an inhibitory effect because by inhibiting that muscle, it tends to cause those air tubes to dilate, which means to increase in size. And of course, that increases airflow, which is exactly what we want during a fight or flight response. The sympathetic division also, for instance, tends to dilate or relax the smooth muscle in some blood vessels in the body. It tends to constrict most blood vessels, but it relaxes and causes dilation in certain arteries in the body, specifically one serving the skeletal muscles, so we can increase blood flow to the skeletal muscles. It also tends to dilate or relax the muscle in the walls of blood vessels serving the skin, so we can shunt more blood to the skin, which helps us radiate heat during the fight or flight response. This is why we tend to turn red during exercise, for instance. And as you can see here, of course, it does also inhibit some other systems that aren't particularly relevant to the fight or flight response, things that you can suspend, activities you can suspend for a short time while you're busy focusing on the respiratory system, the cardiovascular system, the skeletal muscles doing their thing during an emergency. And so it generally, again, has an inhibitory effect on things like the digestive system and the urinary system. It tends to reduce activity in those systems and reduces blood flow to those systems. So again, the fight or flight response, the sympathetic division, simply does whatever is necessary to get the body ready for action. It doesn't always necessarily involve stimulation of its effectors. Sometimes that involves inhibition. The same can be said for the parasympathetic division, which is the other part of the autonomic nervous system. The parasympathetic division is referred to as the resting and digesting system. The resting and digesting system. So this is the part of the autonomic nervous system that has the predominant effect under normal resting conditions. And so at rest, the parasympathetic division is constantly, for instance, tending to slow the heart down. I think I mentioned in an earlier lecture that the heart has an intrinsic rate. It wants to contract at about 100 to 120 beats per minute. But it doesn't. The typical heart rate at rest is 70 beats per minute. This is due to the parasympathetic division, the resting and digesting system, which is constantly putting the brakes on that pacemaker of the heart and telling it to slow down. It also tends to have an inhibitory effect on our breathing as well as on our ventilation rate. On the other hand, it has a stimulatory effect on the digestive system and the urinary system. And so urine production and urination itself tend to be stimulated by the parasympathetic division, and much of digestion, stomach activity, intestinal activity, both in terms of secretion of enzymes and muscle contraction, are stimulated by the parasympathetic division. So again, has both excitatory and inhibitory effects as it does its job, and its job does tend to be opposite, antagonistic to the sympathetic division. As we'll see later, they don't always antagonize each other. Sometimes they work together, but the most common condition is where they antagonize each other, and so one system acts like an accelerator, the other acts like a brake, and it's the interplay between accelerator and brake that gives us the fine control we exert when we drive. Very similar situation in the body with these two systems. If you've been following along what we've been saying, you realize that old saying, that old adage, don't eat and then go swimming right away, is good advice, because when you eat a meal, especially particularly a big meal, that creates a lot of parasympathetic activity. Because we need to digest that food, and so we start sending lots of blood to the digestive organs to help absorb that food, and we stimulate muscle activity in the digestive system and so on. And we just don't have a lot of resources left over for the skeletal muscles. And so if we go into the water and try to swim, that's going to try to activate the sympathetic division, while the parasympathetic division is already working hard to digest that meal that you just ate. And essentially you've now got the two systems at war with each other, competing with each other. And again, it'd be like trying to drive while pressing the accelerator and the brake at the same time. It's not good for your car to try to drive that way. Similarly, it's not good for your body to put these two systems in conflict with each other. Now, if you do this and you exercise at the gym, you might cramp up because the skeletal muscles can't get enough blood due to this conflict. But at that point, you just stop exercising. But if you're swimming, especially in deep water like the ocean, and you cramp up, well, that's a different situation, and that, of course, could lead to some serious consequences. So it probably is not a good idea to eat a particularly large meal just before exercise, because you're going to throw these two divisions into conflict with each other. Here we see a diagram of the central nervous system highlighted in blue, brain and spinal cord again, and then the rest of the system shown in yellow, the peripheral nervous system. And very nicely, again, a table to organize everything we've been discussing. All right, at this point, we're going to start talking about the cells of the nervous system, and there are basically two types of cells that make up neural tissue, and those are referred to as neurons and glial cells. Neurons and glial cells. Of the two, the neurons are what we call the functional cells of the system. They're the functional cells of the system because they are the ones that actually carry out the job of generating nerve impulses and conducting nerve impulses around the body. The glial cells, as we'll see, perform a support role for the neurons. So we're going to start with neurons, the functional cells, and then we'll move on to glial cells shortly. So neurons have three distinctive properties. These properties are not unique to neurons. There are other cells in the body that possess these properties, but these three properties are characteristic of neurons. And when you put all three of them together, that tends to be relatively unique. And I'll explain what I mean by that as we go through it. So first of all, they're excitable, which again, like muscle cells, means they can respond to receive stimuli and respond to stimuli. And again, remember, muscle cells are excitable. When they receive a neurotransmitter like acetylcholine, for instance, they respond. That's excitability. When smooth muscle is stretched or gets cold, they respond. That's excitability. So this idea, this characteristic of excitability is not unique to neurons, but it is certainly characteristic of neurons. The second fundamental property of neurons is conductivity. And what this means is that once they are stimulated, once they are excited, they can respond or react to that excitation by producing electrical signals that are capable of being conducted or propagated some distance and onto other cells. So this is conductivity, this ability for a signal, an electrical signal, to travel along the length of the cell. In our last chapter, we referred to these signals as graded potentials and action potentials. And that's what we're talking about here, this ability to create graded potentials and action potentials. And then, of course, we know action potentials spread like dominoes by each one stimulating the next, and so on and so forth, just as we saw in muscle cell membranes. So again, this is not unique to neurons because we've already seen that muscle cells are capable of this conduction as well. And then the third property of neurons is secretion. And we know, from previous discussion, when action potential reaches the end of an axon, the axon terminal, the axon can respond by secreting or releasing a chemical known as a neurotransmitter. It does this by exocytosis, and so this qualifies as secretion. And once again, many cells perform secretion. Both endocrine and exocrine glands, for instance, perform secretion. However, when you put all three of these things together, you see that you have a fairly unique set of characteristics. So while muscle cells are excitable and conductive, they tend not to be involved in secretion. And while there are many glandular cells that are involved in secretion, they tend not to be conductive. They're excitable. This is how we stimulate glands to secrete. But most glands do not have that conductivity property. So it's this combination of three features that tends to identify or make neurons unique. Now, there are a variety of different types of neurons, and so we can classify them into categories. And there's actually a couple of different ways to classify neurons. We can classify them based on their function, what kind of function they perform, excuse me, or we can classify them based on structure, how they are built. We're going to start with a functional classification of neurons. And in this functional classification, we have three different categories of neurons: sensory neurons, interneurons, and motor neurons. Sensory neurons, which again can also be referred to as afferent, incoming. Sensory neurons are the neurons that do the job of picking up stimuli out in the body, somewhere outside of the central nervous system, and transmitting that information towards the central nervous system, towards the brain and spinal cord. And again, these stimuli can be external stimuli that are out in the environment, or they can be internal stimuli that are within the body itself. Again, things like blood pressure, body temperature, and so on. But sensory neurons do the job of carrying the message towards the central nervous system. Interneurons are neurons that lie entirely within the central nervous system. There is no part of an interneuron that becomes involved in the peripheral nervous system. So while the other two categories of neurons will have at least parts of the neuron out in the peripheral nervous system, interneurons, the entire cell is completely restricted to lying within the central nervous system. What do interneurons do inside the brain and spinal cord? Their job is to make connections. Their job is to pick up the signals coming in from the sensory neurons and distributing those signals to their appropriate destination. Now, we said that one of the jobs of the central nervous system is integration, processing, perception. This is the job of the interneurons. They pick up the signal, and that signal often, not always, but often is sent up into the brain, where various parts of the brain, neurons in the brain, will process that information. This is what's involved in what we call thinking, memory, analysis, emotion. All of these things have to do with interneurons working and communicating with each other in the brain primarily. And so these are the integrative piece of the nervous system, the interneurons. As you might guess, this is a fairly complicated task to do this job of perception, thinking, and integration, and so on. So about 90% of all our neurons are in this category of interneurons. Of course, eventually a decision might be made to respond to a stimulus. And in order to respond to a stimulus, we have to be able to send a message back out to the body. To get a message back out to the body from the central nervous system, we need another type of neuron, and this is the third type of neuron, called the motor neuron, also known again as the efferent neuron. The job of the motor neurons is to send signals, nerve impulses, from the central nervous system back out to the body to create a response. And so again, the target of motor neurons is always going to be either muscle cells in a muscle or glandular cells in either an endocrine gland or an exocrine gland. So So here we see a diagram, again, a visual of what we've just been talking about. We see that we have sensory neurons that pick up a signal out in the body, send it into the central nervous system. Inside the central nervous system, we have all the interneurons that are making connections. Of course, the very simplest kind of connection that an interneuron could make is to simply pick up a signal from a sensory neuron and route that signal directly to a motor neuron, and then have the motor neuron travel straight back out to an effector organ. This very simple kind of pathway, by the way, is generally what we refer to as a reflex. A reflex is the simplest neural pathway that the body can create between sensation and response. For many basic body functions, reflexes are the way to go. We don't want to have to think about them. There are literally thousands and thousands of basic housekeeping jobs that the body has to take care of moment to moment. Blood flow, blood pressure, respiratory rate, hydration levels, blood glucose levels, blood lipid levels, so on and so forth. All these various things that we have to regulate. And literally, we could not simultaneously think about all those things, doing all those things at the same time. And so the benefit of reflexes is to very short, very stereotypical kinds of responses to very predictable kinds of situations, so that we don't at a conscious level have to think about these things. But of course, some neural pathways are more complicated, where we receive information and the interneuron then will send the message, route the message up the spinal cord into the brain, where literally thousands of interneurons can get involved in the signaling, and that is what we would refer to as higher level kinds of thinking, thoughts, emotions, etc. Okay, before we talk about the structural classification of neurons, different kinds of neurons based on how they are built, we have to know a little bit about the structure of a neuron, so then we can contrast the different shapes of neurons. So at this point, we're going to talk about some basic components of a typical motor neuron, because this is the most typical kind of shape of a neuron. But again, we'll come back to that when we talk about the structural classification. So just to orient you, we're somewhere around objective 31 right now in our objectives. So the basic parts of a neuron consist of things like a soma, which is also known as a cell body or a neurosoma, Nissl bodies, dendrites, and axons. These are the four major parts that we'll talk about. And we're going to start with the soma. The soma, or cell body, is where it is usually referred to, or often referred to, as the biosynthetic center of the neuron. So this is where much of the living processes take place. So what you find in the soma, or cell body, are all the things you typically find in a living cell. You've got a nucleus, of course, with the genetic material. You've got most of the cell's cytoplasm, containing your typical organelles, the mitochondria, lysosomes, Golgi bodies, vesicles and inclusions, endoplasmic reticulum, especially rough endoplasmic reticulum, cytoskeletal elements like microtubules and microfilaments, and so on. So the neuron cell body, or soma, has all the typical components of cells. It does have inclusions, little pockets or bubbles of endoplasmic reticulum that contain various materials, glycogen for energy storage, interestingly some melanin and lipofuscin, which are pigments. These build up as lysosomes digest old organelles, and so as individuals get older, their neuron cell bodies, their soma, tend to get darker as we accumulate these pigments. The cytoskeleton is fairly extensive, with many microtubules and microfilaments, and these microtubules and microfilaments tend to break up the rough ER into compartments of rough ER, so separated sections of rough ER. And this rough endoplasmic reticulum, remember rough endoplasmic reticulum is involved in protein synthesis primarily. This compartmentalized rough endoplasmic reticulum tends to pick up stain, preferentially, differentially, and this is what is referred to as a Nissl body. These compartments of rough ER that stain deeply are referred to as Nissl bodies. What the neuron cell bodies lack, noticeably, are centrioles. And recall that centrioles are little packets of microtubules that are involved in mitosis. Spindle fibers of mitosis anchor to centrioles. And so without centrioles, the cell cannot perform mitosis. So this is another kind of cell in our bodies that is amitotic. And of course, the most obvious significance of this is when you damage neurons, if you destroy neurons, you cannot replace them with functional tissue, cannot replace lost neurons with new neurons. So the number of neurons you have right now, that's the maximum number you're going to have for the rest of your life. And if you lose some, you're never going to replace them. All right, so we've seen that the neuron cell body is the main biosynthetic center where most of living processes and metabolism takes place. What about dendrites and axons? Well, dendrites and axons are both extensions, cytoplasmic extensions of the neuron cell body. So they are basically little out-pocketings of cytoplasm, but they aren't necessarily little. They tend to be long and thin. Axons tend to be much longer than dendrites. Now there are several differences between axons and dendrites. So let's talk about dendrites first, and then as we talk about axons, we'll see how they're different. So again, dendrites are cytoplasm-filled branches that come off of the neuron cell body. Dendrites primarily serve a role of receiving signals from somewhere else. It says here from other neurons, so that's the most common case, because again, most neurons are interneurons that are receiving signals from other neurons. But remember, in sensory neurons, the signals that dendrites are going to be receiving will be coming from the body. So their job is to receive signals and carry those signals toward the cell body. Dendrites do not carry action potentials. We described an action potential briefly in the last chapter when we talked about muscles. Remember, we described action potentials as a very, again, stereotypical change in membrane permeability, a flow of sodium ions in followed by a very characteristic flow of potassium ions out, creating a very characteristic momentary change in membrane potential. And those were called action potentials. They are all or nothing stationary phenomena. Dendrites don't do that. And again, it's because dendrites don't have voltage regulated gates. In order to do an action potential, perform an action potential, the membrane has to contain voltage regulated gates. Hopefully you remember this from the muscle chapter. So instead, what dendrites experience as a signal is what we call, again, graded potentials. They have chemically gated channels that can open or close to varying degrees based on how much they're stimulated. And so they produce graded potentials of different sizes that travel towards the cell body. We will come back to this much later in the chapter when we talk about the electrophysiology of neurons and how they function. But for now, since we do understand the difference between an action potential and a graded potential, I think it's useful to point out that dendrites only experience graded potentials. As you can see, what's implied here is that neurons can have more than a single dendrite per cell. some neurons, it's rare, but some neurons have just a single dendrite. The vast majority of neurons have multiple dendrites. And of course, the more dendrites a neuron has, then the more, essentially, the more antenna, the more feelers it has, and so the more information it can pick up from its surroundings. So that's dendrites. They are cytoplasmic extensions that carry graded potential signals toward the cell body, and there may be one to many dendrites per cell. An axon, on the other hand, is quite different from dendrites. Axons are also, by the way, known as nerve fibers. So when we look at a nerve carrying messages to and from the spinal cord, say, most of what you find inside that nerve are just bundles of axons. Axons, first of all, they're also cytoplasmic extensions like dendrites, but they originate from a special funnel-shaped region called the axon hillock. So they're right from their origin, they look different from dendrites because they always originate from a funnel-shaped region protruding from the neuron cell body called the axon hillock. Axons tend to remain unbranched for most of their length. Dendrites can branch very quickly, so a single dendrite can look literally like a tree with multiple branches. Axons, it's not that they don't branch, they just don't branch much initially. Once they get out by their effectors, once they get out to their targets, of course, then they can branch quite a bit. And these are referred to as axon collaterals. And so we remember, and we should already know this, because remember we talked about motor units in the last chapter, and we said a motor unit was a single axon in all the muscle cells that it innervates simultaneously, that it serves simultaneously. So clearly we already knew that axons can branch and serve multiple target cells all at once, because that is what a motor unit is. So these branches are called axon collaterals, and they tend to occur away from the neuron cell body, closer to the effector or target organ. All right, so that's what we mean here when we say the branches are on the distal end. Axons are specialized for rapid conduction of signals, and so the kind of signal that axons experience is different from dendrites. Dendrites, remember, experience graded potentials, which don't spread very fast and tend to peter out as they spread. They tend to diminish in size as they spread. But axons carry a kind of signal that moves very rapidly and does not diminish as it travels. This is the domino effect that we talked about. So this is, again, something we're familiar with. These are action potentials. So axons experience action potentials, nerve impulses essentially, whereas dendrites do not. So we see that axons are different in terms of they originate in an axon hillock, and they carry action potentials, not graded potentials. Another difference that's not talked about in this particular slide, another very important difference between dendrites and axons, is that axons always carry messages away from the cell body. Dendrites, remember, carry messages toward the cell body, but axons carry action potentials, messages away from the neuron cell body, away from the soma. Axons, because they're so important in the function of the nervous system, they tend to get their own specialized terminology. So axoplasm is the cytoplasm of an axon. Axolemma, again, plasma membrane of an axon. I don't tend to use these terms, but I just point them out because some people do prefer to use these terms. A couple final differences between axons and dendrites. Whereas you could have, and usually do have, many dendrites per cell, per neuron, you never have more than one axon per neuron. All right, only one axon per neuron, never more. So that's an important difference. And as we see here, there are some extremely rare cases of neurons that have no axons, and we'll mention those again in just a few minutes. And then finally, some axons have what's called a myelin sheath around them. A myelin sheath is a wrapping of a certain kind of glial cell that wraps itself around the axon. And the myelin sheath has some functional consequences for the axon. One thing it does, for instance, is it speeds up the rate of action potential transmission in an axon. So we're going to talk about myelin sheaths again a little bit later in this chapter, but only axons can have myelin sheaths. Dendrites are never myelinated. Dendrites are never myelinated. Axons may be myelinated. There are some axons that are not myelinated, do not have a myelin sheath, but axons, some axons, and many axons do have myelin sheaths. Okay, so those are the two kinds of cytoplasmic extensions, axons and dendrites, and some of the differences that exist between them. And of course, remember that the very tip of an axon is referred to as an axon terminal. This is a swollen tip of an axon that contains the synaptic vesicles that are full of neurotransmitter molecules. Again, we're very familiar with this because when we looked at the neuromuscular junction, we saw that the axon terminal contains synaptic vesicles full of acetylcholine. I do want to point out here that acetylcholine is not the only neurotransmitter of the nervous system. Acetylcholine happened to be the neurotransmitter of the neuromuscular junction. It's the one we use to stimulate skeletal muscle cells. But it's not the only neurotransmitter of the nervous system. There are dozens and dozens of different chemicals that serve as neurotransmitters in the nervous system. And later in this chapter, we will talk about some of the classes of neurotransmitters. But just for your information, some perhaps familiar chemicals that you've heard of that function as neurotransmitters in the nervous system are things like norepinephrine, dopamine, serotonin, and some other molecules. So I just want to make clear that neurotransmitter doesn't always mean acetylcholine. And of course, it's the axon terminal again that releases the neurotransmitter from synaptic vesicles at a junction or connection with the next cell. And remember, in most cases, these junctions between an axon terminal and the next cell is called a synapse. So the neuromuscular junction, recall, was a type of synapse between an axon terminal and a muscle cell. And again, those aren't the only cells that are involved in synapses. You can also have a synapse, of course, between an axon terminal and a glandular cell, because again, those are the other kinds of effectors of the nervous system. And actually, the most common type of synapse is between the axon terminal of one neuron and a second neuron. And again, this is most common because this is interneurons and how they communicate with each other. And interneurons are they are the most common type of neuron. And most commonly, the axon terminal will communicate with the dendrites of another neuron at a synapse. It doesn't have to be the dendrites, but that's the most common type of synapse between neurons. So here we have a picture of a typical motor neuron or interneuron with the various parts that we've been discussing. Again, the multiple dendrites in most cases that are highly branched, the cell body, the soma, where most of the cytoplasm and organelles are located, a nucleus, usually centrally located. Here we have the funnel-shaped region called the axon hillock, which essentially is the base of the axon, the single axon, which extends from the neuron cell body. Axons, by the way, tend to be longer than the dendrites. Notice that axons do branch, just not as extensively as the dendrites, until you get to the tips of axons, and then you find lots of branches of the axon. Some people refer to these multiple branches at the tips as telodendria, but we don't use that term here. And then, of course, we have at the end of each branch, you have enlarged axon terminals containing the neurotransmitter molecules. This particular axon is myelinated, meaning it has a myelin sheath. And as I mentioned already, a myelin sheath really is just a wrapping of certain kinds of glial cells around the axon, creating a sheath or a wrapping essentially around. Notice it takes more than one glial cell to make a myelin sheath, because each glial cell is much shorter than the length of a typical axon. So you have to align glial cells end to end to end to create the myelin sheath. But again, more on that a little bit later. Here we see neurons under the microscope, and again you see primarily their dendrites radiating out. So they have this very characteristic spidery looking appearance under the microscope. These are so-called giant neurons. These are perhaps some of the best neurons to look at, because you can most clearly see things like the cell body with the centrally located nucleus. You can see the multiple dendrites. Most of these extensions are dendrites sticking out. On this neuron, you can clearly see the funnel-shaped axon hillock leading to the axon. And on this neuron, it's not quite as clear, but this is the funnel-shaped axon hillock leading to the axon. You'll notice sometimes it looks like it has multiple hillocks. And so sometimes it's just a matter of getting used to identifying the single hillock. And yet another look at a neuron, and once again on this one I think fairly clear that we have a funnel-shaped axon hillock leading to one extension that's longer and less branched than the others. That's the axon. Okay, so now that we know the various parts of a neuron, we can talk about neurons from a structural classification perspective. We've already classified neurons based on their functional classification, right? Sensory, motor, and interneurons. Now we're going to talk about them based on their structure. And once again, there are essentially three categories. There's technically four, but the fourth one is extremely rare. But the big three are multipolar neurons, bipolar neurons, and unipolar neurons. So what are we talking about here? Essentially what we're talking about is how many cytoplasmic extensions the neuron has. And so a multipolar neuron would be a neuron that has a single axon, because remember there's never more than one axon. So a single axon and multiple dendrites. This is by far the most common structural class of neuron in the body, because not only are interneurons multipolar in structure, and remember interneurons are 90% of our neurons, but motor neurons are also multipolar in structure. So if interneurons are 90% and then we add motor neurons to that, we see that multipolar neurons make up about 99% of our neurons in our nervous system. So by far, the multipolar neuron is the most common type of neuron. Second most common, but of course by far less common, but second most common is the unipolar neuron. A unipolar neuron has a single process out of the neuron cell body. Now, this is confusing, because how in the world can a neuron have dendrites and axons and yet have only a single process? Well, essentially what we're looking at here is that, and let me draw a little picture for you. You have a neuron cell body, right? And the neuron cell body has a single extension. And so, of course, the question is that a dendrite or an axon? Well, what's going to happen very quickly is that single extension is going to split, and it's going to give rise to an extension that goes in one direction, and it's going to give rise to an extension that goes in another direction. But that still doesn't help us answer, well, where's the dendrite and where's the axon? Well, we have a bit of a problem here, because we're going to have to violate one of the rules we just said about neurons. Whoops, there's the nucleus and some organelles and so on. So let's say that we've got a signal coming in this way from somewhere, a sensory organ or somewhere. This part right here, this part of the neuron is going to pick up those signals. Well, what parts of a neuron pick up signals and carry them towards the cell body? Well, that would be dendrites. So we would call these dendrites. These tips right here, they're receiving signals and going to start sending them toward the cell body. And as is typical of dendrites, of course, they only experience graded potentials, and they are never myelinated. So we have typical dendrites starting right here. Graded potentials instead of action potentials, and we never see myelination in this region. But what happens then, of course, is the signal is now going to be carried toward the cell body. But once it gets to this split point here, it doesn't go up into the body. It simply continues on here. And once the signal arrives here, we're going to get the release of neurotransmitter molecules. So these are clearly axon terminals here. The question becomes then, well, when did it change from dendrites to axon? Well, if we were to stick to what we talked about, we would say that, well, this must all be dendrite. This region here must all be dendrites because it's carrying the message toward the cell body. And it becomes an axon after we pass the cell body. And that would make a certain amount of sense based on what we talked about. The problem is that starting right about here, we see a myelin sheath. So all of this in this kind of neuron is often myelinated. Well, what did we say about dendrites? Dendrites are, we said, not myelinated, never myelinated. So if we call this extension right here a dendrite, we're going to have to violate that rule that dendrites are never myelinated. The other thing to appreciate that what starts happening at this point is we don't have graded potentials anymore. We have, starting right here, we start to experience action potentials. And we said that dendrites never experience action potentials. They only do graded potentials. So we're going to have to violate a rule. Either we violate one rule and say that, and violate the rule that dendrites always carry messages toward the cell bodies, not axons, or we violate two rules. The two rules that we'd violate if we call this a dendrite would be that dendrites don't experience action potentials and dendrites aren't myelinated. So we choose to violate just one rule. The rule we're going to violate is that dendrites are the extension that carry messages toward the cell body. So right after this initial region where we pick up a signal, once we start seeing action potentials and myelination, we call this whole portion of the unipolar neuron the axon. This entire structure is the axon. So instead of the axon originating as an axon hillock from the cell body, the axon simply originates right here as we get a transition and we start to get voltage-gated channels capable of experiencing action potentials. So we go directly from dendrites to an axon. So unipolar neurons are interesting in that regard, in terms of what we call an axon and what we call a dendrite. Unipolar neurons, again, I said, are the second most common class of neuron, but again, very, very rare relative to multipolar neurons. But they are not uncommon because we have, of course, billions of neurons. And this type of neuron is the most common type of sensory neuron in the body. So most of our sensory neurons carrying messages in from the body to the central nervous system are these unipolar neurons. And of course they're called unipolar because the cell body has a single extension. All right, and then the next two are quite rare. Bipolar neurons, as the name implies, have a single axon and a single dendrite. They are involved in sensation in some areas, fairly restricted. We only see them in the olfactory mucosa in the roof of the nasal cavity for smelling. We have some in the retina of the eye involved in the transmission of light impulse or action potentials regarding vision and light to the brain. And some are found in the inner ear as well, where balance and hearing take place. And then the absolutely least most common type of neuron is the anaxonic neuron. An anaxonic neuron has dendrites but no axons. Dendrites but no axon. And these are very unusual neurons with very unusual type synapses, and you just find them, again, a few in the retina, and some in the brain, and then some in the adrenal gland as well. Essentially what happens is these neurons can get stimulated through their dendrites, but instead of having an axon release neurotransmitter, the soma, the neuron cell body, leaks out neurotransmitter. And I think that's all I want to say about those kinds of axons for now. And here we see nice diagrams of the various neurons that we just talked about. Again, the most common class, multipolar neurons with their slew of dendrites and branches, single axon as is typical. Bipolar neuron, very nicely, single dendrite and axon. Unipolar neuron, as I drew in the previous slide, with a single extension that then splits, a mass of receptive dendrites at one end, and then this whole thing is called the axon. And then an anaxonic neuron that contains only dendrites and no axon. Okay, this is a likely place to stop for today. I promised you that I'd try to restrict these lectures to an hour, and I haven't done a very good job of that lately. So we will stop here at objective number 52 for today, and when we pick up next time, we will talk about we will start talking about how we transport materials along axons. Axons can be very long. Axons can be up to several inches or even feet long. And so we're going to need special transport mechanisms to get materials from the cell body out to the axon terminal and vice versa to keep the axon terminals viable. And so that's where we'll pick up next time with objective number 53. Until then, take care.